Good afternoon. It's a great honor to speak here at GBCC 2026. I'm Hyung, a medical oncologist from Cancer for Breast Cancer in National Cancer Center, Korea. Today, I will briefly share recent advances in targeted therapy for metastatic breast cancer. Before we talk about data, let me explain one simple concept. Imagine that each pink cell you can see here represents a normal, healthy cell, and among them, there is one abnormal cancer cell. Traditional chemotherapy works like a white spray. It affects many rapidly dividing cells, both cancerous and normal. But targeted therapy works differently. It looks for a specific feature, a molecular target that is mainly present on cancer cells. Once the target is identified, the treatment can focus precisely on that cell. In other words, instead of treating the entire field, we are aiming at the exact cell that carries the abnormal signal. That is why we call it targeted therapy. It is not random. It is guided by biology. Now, over the years, our ability to identify, to attach these targets has evolved significantly. When we talk about targeted therapy, it is important to understand how the concept itself has evolved. In the beginning, targeted therapy meant blocking a receptor, for example, HER2. Trastuzumab was revolutionary because it showed us that if we block a specific receptor, we can dramatically improve outcomes. Then came dual blockade, combining trastuzumab with pertuzumab, which demonstrated that deeper inhibition of the same pathway could produce even greater benefit, and that small molecular inhibitor targeted signaling pathways inside the cell, expanding our ability to overcome resistance. But today, we are in a totally different new era. Targeted therapy is no longer just about blocking signals. It is about delivering highly potent therapy directly inside the cancer cell with precision. This is the era of antibody drug conjugate, where the antibody acts like a delivery vehicle carrying a powerful payload. So the definition of targeted has fundamentally changed. Let me explain how antibody drug conjugates work. I often describe them as a guided missile. The antibody is the guidance system. It recognizes and binds to a specific target on the cancer cell, like a GPS locating the exact address. Attached to that antibody is a very potent chemotherapy payload. This is the missile itself. Once the antibody locks onto the cancer cell, the entire complex is pulled inside. Inside the cell, the linker is broken down. The payload is released directly where it is needed. So instead of exposing the whole body to chemotherapy, we are delivering a powerful drug precisely into the cancer cell. And modern ADCs also have what we call a bystander effect, meaning nearby cancer cells can also be affected. That is why ADCs represent a major advancement. They combine precision targeting with powerful intracellular delivery. And this guided missile strategy is now transforming outcomes across multiple subtypes of metastatic breast cancer. In the past, we thought of HER2 in a very simple way. Either it is on or it is off. If it was clearly on, patients received the HER2 targeted treatment. If it was off, they did not. But now we understand that HER2 is not just a light switch, it is more like a dimmer switch. It can be bright, medium, or even faint. And even when the signal is faint, modern treatments can still recognize and target those cancer cells. This has changed the way we think about HER2. It means more patients may now benefit from targeted therapy than we previously thought. And with this new understanding, let's look at how antibody conjugates are making this possible. Traditionally, breast cancer treatment was strictly divided by subtype, but ADCs are challenging those boundaries. Because of this design, they can work even in tumors with heterogeneous or low antigen expression. Trastuzumab deruxtecan extends beyond HER2-positive disease. Sacituzumab govitecan works across both TNBC and hormone receptor-positive disease. The message is clear. Biology matters more than rigid categories. In HER2-positive metastatic breast cancer, this new treatment, trastuzumab deruxtecan, made a major difference. Patients were able to control their cancer for much longer compared to the previous standard treatment. This was not a small improvement. It was a big step forward. Because of these results, trastuzumab deruxtecan is now the preferred second-line treatment worldwide. And importantly, its benefits are not limited only to HER2-positive disease. In the previous slide, we talked about HER2-positive disease, but this next study looked at a different group, patients whose tumors had only low levels of HER2. In the past, these patients were considered HER2 negative and were treated mainly by chemotherapy. But this study showed that the same targeted drug helped them as well. Patients were able to keep their cancer under control longer compared to chemotherapy alone. This is important because it expands who can benefit from targeted treatment. So HER2 is no longer just positive or negative. Even low levels can now be treated. And while HER2 targeted therapy has advanced rapidly, precision medicine goes even further, including targeting how cancer repairs its DNA. Let me briefly explain another important targeted treatment, PARP inhibitors. In some patients with a BRCA mutation, cancer cells already have a broken DNA repair system. To survive, they depend on a backup repair pathway. PARP inhibitors block that backup system. When both repair systems are shut down, the cancer cells can no longer survive. I often compare it to a house that has lost its main power. When the emergency generator also stops, the light go out. Normal cells usually still have their main repair system, so they are less affected. This is a precision medicine targeting a specific weakness in the cancer in a smarter and more focused way. This slide shows what happened when a PARP inhibitor called olaparib was compared with standard chemotherapy in patients with a BRCA mutation. Patients who received olaparib were able to keep their cancer under control longer than those who received chemotherapy. What makes this especially meaningful is that the treatment is matched to the biology of the cancer. It works because the cancer has a specific weakness. That is why genetic testing for BRCA mutations is now recommended. It helps us choose the right treatment for the right patient. And while targeting DNA repair is one powerful strategy, cancer growth signals offer another important target for treatment. There is a growth pathway inside the cancer cell that acts like a stroke accelerator pedal. This pathway keeps telling the cancer to grow and survive. In certain patients, genetic changes make this signal even stronger. The good news is that now we have medicines designed to slow down that specific signal. Instead of treating everyone the same way, we look at the tumor's genetic makeup and choose treatment based on what is driving that particular signal. This is another example of a precision medicine, making the treatment to the biology of the tumor. And this approach was clearly demonstrated in a recent clinical trial called Capitol 291. Overall, patients are able to keep their cancer under control longer with capivasertib compared to hormone therapy alone. But the most important finding was this. Patients whose tumors have a specific growth signal change benefited even more. In other words, when the treatment was matched to the cancer's specific weakness, the results were stronger. This reminds us that breast cancer is not the same in every patient. The more we understand the biology of each tumor, the better we can choose the right treatment. This is what precision medicine truly means, not one treatment for everyone, but the right treatment for the right patient. Even when targeted treatment work well at first, cancer cells can sometimes change over time. This does not mean the treatment failed. It simply means the cancer has found another way to grow. When that happens, we reassess and choose the next treatment based on how the tumor has changed. Today, we do not treat metastatic breast cancer with just one drug. We plan a sequence of treatment, adjusting step by step. Our goal is long-term control using different targeted therapies at different times, depending on the biology of the tumors. Targeted therapies have changed how we treat metastatic breast cancer. Today, we have more treatment options than before. We can choose therapies based on the specific features of each patient's cancer. And when one treatment stops working, we often have other options to move to next. Our goal is simple and practical: to help patients live longer and to maintain their best possible quality of life along the way. Thank you for your attention.